All right, we're gonna get started again. Please welcome me. Welcome. Please join me in welcoming Dr. Tom Nichols.
Yes, so of course I'm pinch hitting for Angie Laird, and so I'll be using her slides, but they are kind of community effort, and so that I'm quite familiar with them. We just heard from Angie; she's unwell, but she's fine. I'm sure you'll see her at the meeting. So we've talked about coordinate-based meta-analysis, and now to fill in the other part, which is image-based meta-analysis, sort of where that's coming from and where it's going. Angie has no disclosures, and nor do I. So hopefully after the end of this short talk, you'll have an understanding of the role of image-based meta-analysis in neuroimaging research, including what are the differences between image-based and coordinate-based, what are the advantages of image-based, and then also why we are not all doing image-based right now. Okay, so I think this has already probably been covered already, but just why we're interested in meta-analysis. Of course, there's so many results there in the literature. We need some way of coordinating them, synthesizing them, and especially from the early days of neuroimaging, a lot of the studies are very small, and so you simply need to gather them all together to gain power. And of course, also the hope is that when we combine lots of different results together, the consensus collected result will be more generalizable than any one individual result. So that's why we're doing it in purpose, on general. So where do we sit and how does things work sort of in the general sort of scientific ecosystem? You design an experiment, you come up, you acquire the data, you get the raw data, you analyze this, you get some result, and then you publish. This happens by different people, different sites, different locations, different authors, and then that puts us in a position where we could extract from these papers the coordinates, and then we could do a coordinate-based meta-analysis. And that's what we've learned about already. That's what Kim talked about. But of course, what we'd like to do is the image-based meta-analysis. But of course, that requires having access to the images. And this is actually something I think it's a shame, really. If you compare to the genetics literature, there is a convention that you always upload your Excel spreadsheet of t-values, of p-values for your genetic associations, right? I mean, they've been doing that for decades. Unfortunately, we have not had the same conventions in brain imaging because initially there weren't sort of good standards and there weren't places to share that going back to the early noughts, but of course now we do, and I'll talk about that. But that's one of the reasons why this hasn't been possible. But that is what image-based meta-analysis is, that getting the actual maps of the statistical results, crucially not thresholded, and then combine them into a meta-analytic result at the image level. So I don't have to go through this, but this is the contrast. So the coordinate-based meta-analysis is doing everything just from these coordinates. And honestly, I think it's amazing that you actually get anything. If you think about it, how you can write on a Post-it note these coordinates, really, from each study, right? And then you combine them all together, and you actually get something that's sensible. However, when we went and looked and directly compared, so I don't know if you've thought about this exercise, if you happen to have all the image data yourself, you of course can make the coordinates, and then you could do a coordinate-based meta-analysis. So we did this back in 2009. We actually took a bunch of pain data sets that we had, because there was a pain researcher at Oxford that was doing lots of pain work, and so they had lots of studies where we had all the original data. So we were able to do both. We were able to do an image-based meta-analysis and a coordinate-based meta-analysis. And you can see that, okay, these are not crazy. Okay, the insula, you're finding that with the coordinate-based meta-analysis, anterior cingulate, you're getting kind of the dominant regions here. But you can see there's a whole lot more nuance here, right? There's a lot more detail in the image-based meta-analysis. And what's more, if you're interested in them, of course, we have access to the decreases, right? So in the coordinate-based meta-analysis, only if you've very, very carefully manually curated them will you be sure that the coordinates you have are just for the increases of a task. Whereas when you do a bulk meta-analysis, a large-scale one, you're just kind of getting everything together. So there's just going to be a lot more detail and a lot more sort of precision if we can do the image-based meta-analysis. Oh, I should have a nice little animation there. From coordinates to images. Okay, So again, they are answering two different questions. So in the coordinate-based meta-analysis, you're basically saying where are there consistent locations where these coordinates overlap. Whereas in the images, you're basically saying where are these image-based signals converging and where are they consistent. So there are two sort of useful resources. Well, these are both my articles. Thank you, Angie. So this is a review paper that goes through all the different coordinate-based methods. And then this is a preprint that should be any day. Camille keeps on telling me that she's going to finish this and put this out. But right now it's still available as a preprint, which is a review of all the different types of methods for intensity-based meta-analysis. The funny thing is that what we do right now in brain imaging, where we actually just, so for example, if you are a person who collects your own fMRI data and then you do a group level analysis, well, really, what have you done? You've done a single subject analysis, or you've gotten that you have to fit each subject's data, right, to do the task modeling, and then you combine them. That is a type of meta-analysis. So we actually kind of already do a meta-analysis whenever we combine a group of subjects. And so it turns out if you had access to all the original data, you actually could do effectively a meta-analysis by just using, just like calling FSL or in a certain way. Now the key thing is that SPM can't really do this so well because it doesn't account for the individual variance of each study, whereas FSL does account for the individual variance of each subject, and it actually doesn't care whether those were subjects or other studies, and you can combine them together. But in general, what we do is we just, that's the ideal setting where you actually have the variances from each study, and unfortunately, we don't generally have that. So if we have anything, we're going to have statistic maps from each study, and then we're just going to combine them and just trying to see which areas are showing a consistent change in a given direction in the map. So again, we actually have an ecosystem that can help you do this. So the Neurosynth Compose ecosystem is here to basically help you find and identify studies, but also using NeuroStore, you can actually sort of collect images as well. We have a specification to sort of help you define and create a meta-analysis that is then carried out by NIMAIR. So Julio is here. No, actually, your work comes later. It was another grad student of Angie's that developed NIMAIR, which is a comprehensive Python package that implements sort of basically every conceivable meta-analysis method that has ever been created at the time. But the key thing is that it implements the coordinate-based meta-analysis, but also the image-based meta-analysis. And the Stouffer's one, I think we'll just focus on Stouffer. It's a fancy name. Stouffer's is really nothing more than averaging T-maps. C-maps. That's basically the kind of the most basic way that you would ever want to combine images, and that's what Stouffer's does, and that's what we use. So the thing we're facing against now, if you are going to sit down and say, I want to do a meta-analysis, the problem you're going to find is, of course, the shortage of image-based data. So as I've said, this data has not traditionally been shared. And this is a problem because initially, in the early noughts, we did not have a place to share this data. For the past well over 10 years, we've had this resource called NeuroVault, which is a place which I heartily encourage you. If you are someone who creates your own and publishes your own task fMRI studies, please upload your images to NeuroVault when at the time of submission. You get a link, a permanent link that you can then associate with your paper, add into your paper, and then other people can have access to your statistic maps. And even if you really don't want to use NeuroVault for some reason, put them on OSF or some sort of public fixed long-term repository so they're accessible to other people who then want to do meta-analysis on them. So fortunately, the NeuroVault collections are growing, and you can see this over time, but it's not keeping up with all the literature that's being added. You can add metadata, and we encourage you to add metadata to like what kind of task was this about. So you can add a lot of things. What was the sample size? And this is giving you an idea of what sort of tasks are being uploaded. So because people have taken the time to classify what is the task that's been uploaded. So social judgment, decision making, go no go, monetary delay. So these are the number of images, and of course, there are collections and there are images. So collection is what you define. It's like a repository, and of course there can be multiple images inside that collection. So also what you can do with NeuroVault is you can associate a PubMed ID or DOI with your paper. So that is really, really useful. So then we can, when we see these maps, we can say, okay, what is the peer-reviewed publication or just archival publication that's been submitted and that can be connected to. And the IBMA is very straightforward to do in NIMARE. This is just showing you how easy it is to do a coordinate-based meta-analysis, and likewise, you can do a IBMA meta-analysis. There's Mooney and Laird. This is another variant I didn't mention, but it's if you are so lucky to have your both estimate and your standard error maps, you can use this. This is kind of the best practice, most optimal meta-analysis method for image-based, from intensity-based meta-analysis. More typically, you'll be using the Stauffer's method if you only have the Z-maps available. Okay, so now I can get to Julio's work. So Julio has done the exercise of seeing what can we learn from the images that are on NeuroVault. So he went through the exercise of basically saying, let's try to find some useful data that we can actually go and extract from NeuroVault images, which we'll then submit to meta-analysis. So starting from over 200,000 possible images, we only wanted to take the fMRI bold one, so that's 165,000. We don't want single subject results, we want group level results. So that brings it down more. We want to get rid of the probably really noisy results with the really small sample sizes, so we check out the ones that are less than 10. We only are going to work with T-maps or Z-maps. It is possible people have uploaded F-maps. There's nothing wrong with that in principle, but you just need you need more information to know how to interpret an F-map. And it's not really going to be easy to map and be compatible with a T-map. Of course, if they're thresholded, that's bad. The whole point here is that we want to be able to combine information across all the studies at every single voxel. And if they're thresholded, then that's like a missing data problem. And worse, it's a structured or a meaningful missing data problem, because of course it's when they're too low that they're missing. So get rid of the thresholded images. Also, sometimes you have cases either just through acquisition problems or because it was just a study that maybe it's a study that only looked at the visual cortex, in 7 Tesla and just imaged the back of the brain. That obviously is not going to work to be compatible with a whole brain analysis. So we make sure that every study that we had at least 40% coverage. And then finally, we want to make sure that they have been put into the MNI space, because if they're not standardized or if they're in some funky other space, or I guess it could be cortical surface, then we couldn't do that. So that finally brought us down to about 6,400 images. Okay, so then with that said, we then did classify them by the different type of study that they're in. And what we did is that we said, we will use the Human Connectome project, for which we have the data fully available and it also is on NeuroVault, to do what we can pretty sure is a high confidence meta-analysis for a high confidence result for, not meta-analysis, single study result for working memory, motor, pain, motion processing, and faces. Well, I can see, sorry, the sources are slightly different, but for faces, shape is motor, and working memory, we get that from HCP. And for the others, we get them from pain, from neurosynth. And so these are the results of doing an intensity-based meta-analysis, where we, of course, excluded, in these cases, the HCP, because, of course, we don't want to, double-dip or cheat. And so what you can see is that actually we are getting sensible results. Now, there's a lot that went into this, and I encourage you to look at Julio's preprint, because there are different choices you can make about including studies or trying to find ways to deal with outliers and strange studies. But in general, what this is saying is that we are getting sensible results from the studies that are available in neuroscience, and they're doing, maybe except for the emotional processing, which had a very, very weak result. The other thing that I'll note is that when you do a meta-analysis, if you ever have done a meta-analysis for general clinical trials or outside of imaging, they stress the importance of working with meaningful units, right? So a z-score is not a meaningful unit. I mean, in particular, all things equal, if you could make your study grow and grow and grow, your z-score should get bigger and bigger and bigger for where there's a true signal, right? So that's not a meaningful unit. That's not something that's meaningful in the population. What is meaningful is a Cohen's d, right? That's a standardized effect size. So in fact, one of the first things we did here is convert all of these maps, these T maps, with knowledge of the sample size, convert them into a Cohen's d. So that's why these things here are all actually, the fact this is dimmer here, this is telling us that there was a weaker effect size. We found a weaker effect size in emotional processing, social cognition. One of the reasons why we have such solid and decent is that these are strong tasks, clearly. But it turns out there's also just a lot of studies that have high N with that contributed and give you a good idea of where the working memory effect and where the motor effect is. So this is just some of the results to look at the similarities. So that was the pictures comparing what we found to some references, either HCP or Neurosynth. But just to say, there is the issue of how do we decide which images to include in these analyses. And Julio did the manual work of just individually checking each one. Does this make sense? Is this a sensible study to include? Versus a heuristic of just sort of seeing, is there a positive slope or is there an outlier? Does this study look kind of similar to others, or does it seem like it's an outlier? And what we're finding here is that this heuristic solution is giving pretty similar. In fact, so with working memory, we get improved similarity between the result from the IBMA and the reference by doing these selection, either heuristic, and the best one here was manual selection. And it turns out for some of them didn't make such a difference between motor pain, but emotional processing made a big difference to do the filtering. And the good thing and the result from this was that the heuristic selection, which of course can scale up, you don't have to use your eyeballs on every single one, did a pretty decent job and also sort of improved the similarity between the IBMA result that we got and the reference method. So that's where we are. So really, in an ideal world, we all would have been doing image-based meta-analysis for the last 20 years. But because none of these things, the images have not been routinely shared, we're not able to do that. So Julio, your paper's already up. Yeah, so yeah, look for Julio Perez and Angie Laird. If you search that in Google Scholar, you'll find the recent preprint that's been posted. We'll be developing a workflow that we'll share that it will show you the workflow that Julio developed for selecting and doing the IBMA. In Neurosynth Compose, we have the ability to do index and search right now, and this will allow us to do image. So I'm actually not sure what the state of these. Is this right now?
Index and search?
Yeah.
Yes,
And you can do image selection. I mean, you can work with images in Neurosynth Compose. I mean, that's, or is that to come?
I guess doing the extraction thing is not implemented.
Well, there's nothing to extract, right? Well, you'd have to extract it from NeuroVault. Yeah.
So it would be, that is not implemented.
Yeah. Okay. So these things are coming that we will then bring the work that Julio's done and put it into Neurosynth, and basically extend what is not there are basically detailed workflows for IBMA, and that's something that we will add to Neurosynth. Okay, I'll stop there. Any questions?